Oops, now I have to see. Okay, I would like to start with a slide on where AI can today help pathologists. And maybe let me first point you to what we are doing in pathology in routine diagnostics. So we are basically looking at the images you see in the left of this slide. So these are histological specimens that are stained with the standard hematoxylin eosin stain, rendering these pink and blue images. And looking at these images, we can say whether a patient has cancer or some infection or another kind of disease that is changing the tissue structure. So it is, of course, obvious that AI may help with these image analysis tasks. And as you can see here in this image, AI can relatively easily detect the lung cancer contained in this image, which is highlighted here in this heat map, and all the yellow, orange, and red pixels are a precise identification of the lung cancer in this tissue. At the same time, one has to say that this is a relatively easy task. And as you all know, the current limits of AI approaches consist basically in the fact that you need a lot of training data and that then the AI can interpolate pretty well, but that extrapolation, something the human brain can do relatively easily, even with a limited training data set, the AI can't do. And at the same time, this is really a fundamental limitation at the moment, particularly in medicine, where you are facing a lot of diseases, and some of the diseases are very frequent, as you can see here in the skewed distribution on the left. That is stomach biopsies, and there's a handful of diagnoses that are very frequent. Then you have a lot of very rare diagnoses. So this long tail poses a problem, of course, to the fact that you need a lot of training data, because even if you put together international data sets, it's very difficult to get sufficient numbers of cases to train an AI to really also being able to detect the rare diseases. So for this, to address this challenge, anomaly detection approaches can be of help, and we have started developing these, as you can see here, in a colonic biopsy specimen, where it is, when looking at this, very easy to overlook the actual pathological information. And anomaly detection can help you here not to solve the case, but to point the pathologist to the area of interest. And so you get these heat maps, and here in the green and yellow colors, you can see that some regions are highlighted while the majority of the image is considered as normal, which you see here indicated by the blue shades. And now if you look closer into that region where you have something interesting found by the anomaly detection, you can see here in the upper part that something looks different, right? And that is something that is very rare in this kind of biopsy. So this is actually a metastasis from stomach cancer going into the colon. And there are so few cases of it that it would be practically impossible to train your AI to really clearly detect this as stomach cancer. But with the anomaly detection, you can highlight this region and then have an expert pathologist further review the slides, making it easy not to overlook such an important finding here. Then the second challenge, which you have heard a lot about by Klaus and others during the previous talks, and that's why I don't want to go into details, of course, but point to the aspects where explainable AI or the black box challenge poses a problem in medicine. And that is, of course, in diagnostics, where if you have a black box algorithm, you have the problem that physicians are not enabled to verify the AI result. And second, if you analyze data for research purposes, you may overlook the actual important finding and can overlook certain important biomarkers, for example. So here, of course, opening the black box with explainable AI can be of great great importance and can really help a lot. And this image here on the left, which is a histopathological image of breast cancer, where you would, within Blackbox AI, just get the label that cancer is in the image, and then applying the explainable AI approach you've heard about before can then provide you with pixel precise information on where the cancer cells are located, making it very easy for the pathologist to quickly verify that the AI has correctly classified this image. Apart from just applying this to morphological image-based analysis to identify certain objects, you can also now combine the object detection approach by explainable AI with molecular profiling approach, where you normally have only bulk information. So you get for each of the images, and not with the spatial information, but just for one image, for one patient, you get a certain molecular profile. Molecular profile is important for getting information on how to further treat the patient. So it's really important, but at the same time, very laborious and very costly to identify the molecular profile of a patient. So in this work here, we have tried now to apply explainable AI to train the AI to predict certain molecular properties just based on the image data without any molecular technology analyzing the data. And then also you can get these explainable AI generated heat maps, where now on the right you see in red the cancer cells, in green cells of the immune system, and then in blue the information provided by the explainable AI pointing to the amount of the molecular profile present in the image, and at the same time inferring which image properties the molecular information is associated with. And that you can see in these small tiles here. So you have certain molecular properties that are associated with the tumor cells, then certain molecular properties that are associated with the TILs, the tumor infiltrating lymphocytes, so some sort of immune cells, and then also certain properties that are associated with the stroma of the tumor. While the previous slide has really shown you some work which is focused on research at the moment, we are already now also applying the image analysis, AI-based image analysis approaches to routine diagnostics. And here you can see again an image of a lung cancer, and in the lower part you see again this pink and blue standard stain, which we use in routine diagnostics. And then with the AI, you can easily now highlight the cancer tissue in red and the stroma in yellow, and some necrotic dead tumor tissue in blue. And you can even in a more detailed fashion analyze this on a single cell basis. And here you can see now, just based on this H&E, this pink and blue morphological image, you can now predict in a more detailed fashion what kind of cells are contained in the tissue. And in red, again, you see the tumor cells, but then now you see the other colors in yellow and cyan and blue and so on. You see the tumor microenvironment and here the immune cell composition in the tumor microenvironment, which is currently of high interest for novel therapies, so-called immunotherapies, that target certain aspects of the immune system to fight the cancer, which we'll hear about in the further part of my talk. And you can see that with training the AI, you can even outperform the precision of the pathologist. Here you see the AUC values for the different cell types and the pathologist versus AI. And I said this is already applied in practice, and this is something I briefly want to touch upon. As you may know, and as I alluded to in the previous slide, it is becoming more and more important not only to look at the images of the tumor, but also profile the molecular properties. So look at mutations gene mutations in the tumor or the protein composition of the tumor. So we do a lot of molecular analysis, and to prepare for molecular analysis, you need to get an account of how much tumor is contained in the tissue. And that is normally done by eyeballing the tissue, which is naturally very imprecise. And here we are now applying the AI to precisely quantify the tumor cellularity in these images. As you can see here on the right, in red, you see the tumor cells, and in blue, the normal cells, mostly immune cells. And on the left plot, you can now see the comparison in this cohort where the pathologist did manual eyeballing and what the model estimates. And you can see here in this image, the pathologist eyeballed this as 80% tumor cellularity, which I think you can easily appreciate that is an exaggeration, and that the model estimate here of about a bit less than 20% is much more accurate. And another example here where it's vice versa, the pathologist estimated a low tumor cellularity and the model made a bit over 40% tumor cellularity, which if you look at this heat map here, I think is easily visible that is the more accurate estimation. And this is important to then be able to interpret the molecular analysis results. And here you see the sequencing results of a handful of cancer-related genes, which are important also for clinical decision making. And you can see here that when you normalize the tissue content, the tumor tissue content, you can get much better allele frequency estimations. So basically, you can measure then or normalize to what extent a certain gene mutation is present in the tumor. And this information is important for the treating physician to know whether a patient can benefit from a certain drug or not. So here, AI can help you improve the process of making a more precise, more accurate diagnosis. Then a challenge which may to this audience seem obvious, right? You have to use state-of-the-art approaches and also be really aware of having balanced data sets, multicentric data. And I want to briefly point to this Nature Medicine paper, which is one of the most highly cited papers in application of AI in histopathology or in cancer diagnostics. And here I want to point to this EGFR mutation here, highlighted in this red box. So this is one of the most important molecular properties in lung cancer. And you can see here that the paper claims to be able to predict the mutational status of the EGFR gene with over 82% AUC, right? And so we were trying to re-implement that in a cooperation with a colleague from Heidelberg to see whether the approach published in this paper generalizes also to other data. And we found a substantial performance decrease and were wondering initially whether we made a mistake, but then finally found out that some very simple mistakes were made, such as training on cases also used for evaluation. And basically we corrected this and then found a substantial drop in the AUC from 82% to less than 70%, and at the same time having a substantial increase in a standard deviation. So what we set out is to train and develop models that generalize better. And for that purpose, I want to briefly present a work done by a PhD student, supervised by Klaus and also co-supervised by me partially. And Miriam, she worked on data sets we received from a large breast cancer study group, the German Breast Group, from which we received trial data from 3 neoadjuvant clinical studies, very large studies with several thousand patients. And we set out to predict a certain molecular property called the HER2 status in breast cancer, which is also relevant for therapy decisions. you can basically have a drug targeting this HER2, and then patients benefit from this drug if this molecular property is found in their tumors. And so basically what Miriam did is train a model to differentiate between HER2 positive and negative tumors. And I'm directly jumping to the results to show that here, as opposed to the previous study I showed you, we managed to now train a model that generalizes relatively well, even on a totally independent study. You can see here there's only a minor drop in AUC from 81 to 80%, right? And then resulting in this ROC curve here. And this is also one point I want to mention here, that in many cases, people say, look, if an AI can only do such an ROC curve here, that is not sufficient for routine diagnostics, right? Because we want to get over 99% accuracy. But then you have to always be aware of the fact that all existing diagnostic approaches, and this is here from another paper where they tested established biomarkers in routine diagnostics, and you can see just by eyeballing the two curves that the ROC curves look pretty similar, while the right curve, the AI curve, would be considered as insufficient, while the one on the left is fully accepted in routine diagnostics, right? So I think this is also important when discussing what can AI contribute to routine diagnostics, that we shouldn't be too strict with AI and at the same time being too lax with conventional approaches. So moving one step further, while in the previous examples I've shown you approaches where we wanted to predict a molecular or an image-based property and identify certain objects, quantify tumor cells. But then the next step would be not to predict some of these meta information, but really go to the point what we actually want to know, and that is whether a patient survives or lives longer with a certain therapy or without, because that is the ultimate question we want to answer in diagnostics. And here we had a cooperation with Bristol Myers Squibb, who provided us again with clinical trial data from 3 immunotherapy trials. So immunotherapy, again, as I mentioned initially, is a novel domain of oncological therapy where you can give the patient certain drugs that activate the immune system to fight the tumor, right? And so here the question is, can we, just using, again, our conventional images, can we predict the response of the patient to the therapy based on the histomorphology, based on the pink and blue images you see here on the left? And so for that, we developed a novel approach to learn image embeddings via self-supervised learning that result then in certain clusters of similar morphology. So basically here you see the image patches from the histomorphological slides. And I think even without being a pathologist, you can see that the 4 large squares contain small tiles that look in a way more similar among each other than across the tiles, right? And so these clusters were proposed by the self-supervised learning and then further refined by manual evaluation by pathologists. So we proposed these 50 different clusters and can now for each patient compute these slide histograms. So basically a different composition of the different morphological properties for each patient. And that results in kind of the bag of histopathology words and representing and the histograms representing the histology dictionary. And so using this, we then managed to now improve the prediction of whether a patient's response to this immunotherapy. And the immunotherapy is normally decided or patients are stratified normally by the presence of this marker PDL1, that is programmed death ligand 1. And whether that is present in a tumor normally helps the oncologist and the pathologist to say whether a patient will respond to a therapy or not. But you can see here with the AUCs that the orange line alone, or the orange dots here for the different trials we were trying this on, is not really very good, right? It's around 60% AUC. But now if you add to the PDL1 status, if you add the AI, the features derived with our AI approach, you can see that you can consistently improve the prediction accuracy. And while that is not a black-white difference, so it's really a gradual in improvement, this for clinical trials may well decide whether a trial is successful or not. So here on the right, you see the conventional Kaplan-Meier plot, where you can see the survival of patients after time. So in the blue line would correspond to where the decision was rendered by the clinician or by our AI approach that patients have a better prognosis, and orange means they have a worse prognosis. And you can see that when you AI augment the approach, that the curves separate further. They lie a bit further apart. And so that means that the stratification using the AI approach is improved as compared to the conventional approach. And that may, as I just said, that may well make a difference whether a large clinical trial that costs $1 billion or drug development, whether that fails or whether that is a success. So now moving in the last part of my talk, I'd like to move away from the pure image-based analysis of AI, which is, of course, one of the major domains in pathology, but moving to molecular analysis. As I mentioned initially, we are more and more in diagnostics relying on molecular analyses. And here is an example where we were wondering, can we differentiate between patients who have a cancer in the head and neck region and a simultaneous or metachronous lung tumor? And the problem is here that patients with these head and neck tumors and lung tumors fall in the same risk category, right? If you smoke a lot, if you drink a lot, you have higher risk for both of these tumor types. And then many patients who have a head and neck cancer, so cancer in the head and the mouth, for example, so they often develop metastasis to the lung. And at the same time, they may develop a second lung cancer. So they have two separate tumors, right? And that is a totally different clinical situation. In the case of a metastatic disease, patients will ultimately die and you need aggressive radiochemotherapy, while if you have a second lung cancer, which is detected early, the patient can be cured. And there's no currently available way, or up to this point where we published it, there was no way of telling the difference. So over 84% of the cases remained uncertain or unknown, and basically clinicians had to flip a coin to decide what therapy the patient would get. And so what we relied on is a combination approach of AI and so-called DNA methylation profiling. So DNA methylation profiling means that the DNA has some methylation groups, so some modification on the DNA that tells us where the cells that contain this methylated DNA come from. The patterns of the methylation are highly organ-specific. And so you can see that here in this T/E plot, right, that the head and neck cancer in blue and the lung cancer, that they kind of separate, but it's not a trivial separation. So we trained different classifiers to see whether a proper AI approach can help us classify these tumors. And with a conventional random forest approach, we get an accuracy of about 88%. And then if we trained a neural network, we get over 96% accuracy. And if we thresholded with the classifier scores, we get over 99% accuracy for over 92% of the cases. So basically from a flipping the coin situation to really high diagnostic accuracy with a proper combination of AI and molecular analysis. And that is something we are already applying in routine diagnostics, and that has changed the diagnostic situation drastically. One more recent paper here in Nature Communication, also together with a large international group and Klaus on the machine learning side, we collected data from all over the world on a very rare tumor type, so-called sinonasal tumors, that are also very difficult to diagnose and that have a very bad prognosis. And so we applied a similar approach here using DNA methylation, and you can see that already clusters the tumors according to their conventional diagnosis. You see here the similar or the same colors. Clustering means that the methylation clustering is consistent with the conventional diagnostic approach, but then you see certain clusters here where the colors are very mixed, right? And these clusters here, basically, we use the DNA methylation analysis approach to further look into the molecular properties. And I'm sparing you with the details, but here we did some mass spectrometry-based proteomics on these groups and also sequenced the tumors and then identified 4 novel entities. So each of these circles here correspond not to a mix of the different entities, but these were simply incorrect diagnosis. And each of these circles here correspond to these new subtypes. We were able here by a proper or by a clever combination of AI-based analysis with different molecular properties. We went to proposing novel tumor subtypes, which, as you can see here, have high clinical relevance as the prognosis of these tumors differs substantially. And this is also made available online, so people from all over the world who have such tumors can now do the DNA methylation and then upload the data to our website and get a diagnostic result. So the final few slides, I want to touch upon where we can use these AI approaches in a mainly still research setting. And this is one very interesting new domain or relatively recent domain of molecular diagnostics or molecular technology, that is single-cell sequencing, where we can now, even if we have a limited number of patients and therefore limited training samples for AI, we can now, with the single-cell technologies, sequence about 10,000 cells for each patient. And here you can see from a recent publication from my group, where we sequenced using this approach 12 different patients and received over 120,000 vectors, each containing over 2,000 entries for RNA expression. And this UMAP here, you can appreciate that the different patients kind of cluster sometimes differently, but then there's no trivial separation. And now using the explainable AI approach, using LRP, we can now take certain molecular properties and try to predict other properties, and then again take certain properties and again predict other properties, and then systematically permutate that and use LRP and then infer the network topology of these cells. And you can now see here for four patients, you can now see we can do the sequencing for the tumor because we have individual cell sequencing and the other normal cells. And if we look further into that, we can now identify properties that are specific to the normal cells here on the left, and properties that are specific to tumor cells, and also properties that are specific only, and I'm just jumping for time reasons to the next slide, that are specific to certain subsets of tumor cells. And if we look at the first patient here, patient 18, you can see that if we take these 6 tumor-specific networks inferred by our LRP approach, we can now see that this patient here, and each of these gray dots here is one tumor cell, that the patient has different subtypes or different subclones of tumors. So here you see a certain numbers of tumor cells that are enriched for this pathogenic network T5, which you see here. But then you see also a small group here which is enriched for this network T2. So it is becoming obvious by this that if you have a therapy that targets effectively this pathogenic network T5 here, you may be missing the tumor cells here that are enriched for this network T2. And you can find similar examples for these other patients here. So using this approach, now we can not only better understand the mechanisms of the tumor as a whole, but also dive down deeper into the individual tumor cells, therefore identifying at an early stage, identifying potential mechanisms of resistance to the therapies. And then this may well lead to proposing novel combination therapies where you are targeting at the same time this network T5 here and network T2, so you can hit hard on the tumor without allowing it to relapse and develop resistance to a single agent drug. And with that, I want to conclude where we are moving and what direction we are moving, and that is not only using AI to analyze the individual data modalities, but really combine the different data modalities into one multimodal approach, integrating all available clinical and molecular data, and then in this particular example here, trying to predict relapse in lung cancer patients. And so to conclude what I've shown you, what we can do today relatively easily is identify simple properties in tissue. Yeah, you can easily identify lung cancer, but it's still challenging to identify rare diseases. Then the next and already also partially existing approaches is improving the diagnosis, right, by, for example, the DNA methylation analysis. But then in medicine, AI will show whether it holds up to its promise when we manage to integrate all the different data and come up with comprehensive models that then can make better predictions on how to treat and ultimately cure patients. And with that, I thank you again for the invitation, the opportunity to present my work, and I'm happy to answer questions. Thank you very much.